So the next speaker is Dr. Sam Horn, and he will be introduced by his mentor right here at Sinai, Gareth John.
Good afternoon. I'm here to introduce Sam Hong, and also a slight shift in focus away from pure neuroscience to neuroimmunology, so the interaction of two different systems. Sam completed his undergraduate work at Columbia, where he was Phi Beta Kappa and summa cum laude. He then completed a pre-doctoral fellowship at NIH, which featured a first author publication in the New England Journal of Medicine. From there, he proceeded to the MSTP program at Harvard and MIT, where he studied early patterning of thalamic and cortical visual maps. So that molecular biology has stood him in good stead since he's arrived at Mount Sinai as part of the neurology residency program. He's been chief resident, and during that time he has collaborated with us in the Corinne Goldsman Dickinson Center for Multiple Sclerosis to examine mechanisms that control lesion formation in inflammatory conditions of the central nervous system, particularly autoimmune demyelinating conditions such as multiple sclerosis. In multiple sclerosis, encephalogenic T and B cells drive the formation of aggressive multifocal inflammatory demyelinating lesions, which result in loss of function, paralysis, and blindness. Sam's been investigating the mechanisms by which the encephalogenic cells enter the CNS and the mechanisms that control lesion extent. He already has two publications with us in two authorships in PLOS biology and in brain, and the work he's going to show you today is being prepared for submission probably in about 6 to 8 weeks from now. So I'll just hand over to Sam.
Thank you, Gareth. Thanks to the Leon Levy Foundation and to Dr. Nestler for inviting me to speak today and represent Mount Sinai. So as Gareth said, I'm going to shift gears a little bit from learning and memory to neuroinflammation and inflammatory disease in the CNS. So the title of my talk is Astrocytic Claudin-4 Limits the Size and Severity of CNS Inflammatory Lesions. And basically what I'm going to talk to you today about is a type of cell in the brain called astrocytes and how it modulates the severity of inflammatory lesions and diseases like MS. And I've subtitled the talk A Tale of Two Barriers, and I hope you guys get the reference, and I'll sort of bring it in at the end. It's an analogy that I think is cute and useful for looking at our work. So our lab is interested in multiple sclerosis. Multiple sclerosis is an autoimmune disease that is characterized by relapsing remitting attacks in the central nervous system. It's the leading cause of non-traumatic neurologic disability in young adults in the US and Europe, and it affects about 2.5 million people worldwide. The etiology of multiple sclerosis is still unknown. While we have a lot of therapeutic options and therapeutic approaches to MS, the pathophysiology of the disease is largely a mystery. There are things we understand about it. Clinically, we know that there are two phases of multiple sclerosis. So the most common type of multiple sclerosis is the relapsing remitting phase of the disease. This is characterized by flare-ups or attacks where people get a focal inflammatory lesion either in the brain or the spinal cord, and it leads to a clinical deficit. And the natural history of an attack like this is it comes on over the course of days or a week. It reaches its peak in severity, and then the symptoms slowly resolve. And the reasons for this are really something we're still working on understanding. There's a second phase of multiple sclerosis, which is the progressive phase. And this is characterized by longitudinal, gradual progression of neurologic disability. And what we know from autopsy autopsies of the brains of patients with progressive disease is that there are chronic simmering levels of inflammation, but you don't see the focal flare-ups that are characteristic of relapsing disease. How the two phases are related is unknown. The time course in patients who are untreated is that they'll typically start out with the relapsing phase of the disease and then gradually develop the progressive component. So this is just a graphic showing you the symptomatic course of the disease. People start out in this relapsing remitting phase where they get neurologic attacks and then they have recovery back to their baseline. With time, they may have recovery to a level that's not where they were before, and they can accumulate disability in a stepwise fashion. And then later in the progressive stage, it's really a slow, gradual progression of neurologic disability. So this is the driving force of our work, and the area of multiple sclerosis that we're examining in depth is how inflammatory lesions of the central nervous system form, what precipitates an inflammatory lesion. And a major component of this is actual entry of inflammatory cells and inflammatory soluble factors into the brain and the spinal cord. Now, the brain has classically been understood to be an immunoprivileged organ. There's a barrier between the bloodstream and the brain. And this is quite useful because the brain is a very complex organ, and you want to keep out toxins, unnecessary inflammation from entering into the parenchyma. And broadly speaking, there are two routes of entry into the brain and spinal cord from the bloodstream. The first is what's classically known as the blood-brain barrier, and this is a structural barrier between blood vessels and the actual parenchymal tissue of the brain, and so that's what's noted first. Then there's a secondary point of entry, which I'm not going to get into, and this is the production of cerebral spinal fluid. And this is, these are specialized ports of entry in 4 locations in the brain where the bloodstream is separated by a layer of ependymal cells, and they allow for the production of the cerebral spinal fluid, which is the fluid that sort of cushions the brain. So that's a second possible point of entry. The figure I have here is an immunohistochemical stain from our lab, which shows the blood vessel stained in red, and these green cells here are the astrocytes. So these are supportive cells in the brain parenchyma. So we study how these astrocytes modulate entry of factors from the bloodstream into the brain parenchyma. And so moving a little bit more in detail, if we take a cross-section of the blood vessel and the surrounding cells around it, what we find is there are 2 different layers. So the first layer is the endothelial cell layer, which is actually the blood vessel cell. And so inside the lumen is the bloodstream. And you have the blood vessel endothelial cell, which is here in red. And that's what's classically been recognized as the blood-brain barrier. So these cells, the capillary cells of the blood vessel, are tightly bound to one another, and they very exquisitely regulate the entry of cells, of soluble factors like immunoglobulin, and they protect the brain tissue, the brain and spinal cord tissue, from circulating bad stuff, essentially. Outside of the capillary wall are these end-foot processes by the cells that I pointed out in green, and these are called astrocytes. And they're called astrocytes because they're star-shaped, and classically they've been understood to be sort of supporting cells of the brain. People really didn't pay much attention to them, but what we do know is that they extend end feet that surround the capillary wall. And then between the blood vessel and the astrocytic end feet are another type of cell called parasites, and these are actually being studied actively now, and they're beginning to be understood to modulate the tightness of the blood vessel wall, and they also instruct the astrocytes to bring their end feet into the area. So the parasites play an important role in setting up the structure of the perivascular unit. Again, why is studying the blood-brain barrier important to diseases like multiple sclerosis and to drug delivery? So multiple sclerosis is a paradigmatic example of inappropriate breakdown of the CNS barrier. And these barriers are important to regulate the entry of immune cells and humoral factors. And like in all of medicine, it's a balance between fighting off infection, actually allowing immune cells and immunoglobulin into the tissue when you need to fight off infection, and preventing an overexuberant response where you get auto attack of your tissue from your immune system, which is the autoimmune damage. And work on the blood-brain barrier is also really important because of the principles of drug delivery. And this has been an ongoing challenge in terms of delivering therapeutics into the CNS, is getting things past the barriers to entry. So here's briefly a figure of the pathogenesis of multiple sclerosis. What we do understand is that in the relapsing phase on the left, you get plaque formations, which are focal lesions. There's blood-brain barrier breakdown where the capillary wall breaks open, and you get inflammatory cells, which are these multicolored round cells coming in, and immunoglobulins, cytokines, soluble factors that rev up the inflammation. And then in the long term, you begin to get degeneration where the myelin sheaths around the cells really don't regenerate. You get a denuded axon. The axon starts to degenerate. And these processes that trigger them are an active area of research. Just briefly, these are the types of cells that are coming in through a broken CNS barrier. On the left, MS was classically associated to be a helper T cell disease because in human pathology, those were the types of cells that were most present in lesions. But we know that a lot of other players are affecting MS, and it's probably kind of a symphonic network of signals of these cells collaborating together to induce and perpetuate these lesions. So the cells that have been clinically recognized to be very important are the B cells. These are the cells that produce antibodies, but they also produce a lot of horrible cytokines that can exacerbate the disease. And we know clinically by depleting B cells that people have fewer relapses and the disease is quieted down. And then at the bottom are the microglia, which can be antigen-presenting cells, and they also secrete a lot of the inflammatory cytokines that precipitate lesions. So we already went through the neurovascular unit, but at the heart of the CNS barrier, what actually mediates the barrier at the level of the blood vessel has long been recognized to be the tight junction. So a tight junction is the molecular structure that mediates any kind of barrier throughout the body, and it's evolutionarily conserved. We see it in the gut, in the skin, in the bladder, and we see it in the blood vessels of the brain. And so tight junctions are well characterized, and we know that they're molecular complexes formed of different protein family members. The three major ones are the claudins, the junctional adhesion molecules, also known as the JAMs, and the TAMPs. This stands for tight junction associated Marvel proteins, but these are the tricellulins, occludins. So there are three major families of tight junction molecules. They all come together and they form this complex where they bind cell walls together and they form this kind of impenetrable structure. So we know that they're binding together the capillary wall of the blood-brain barrier, and that's been long studied. And so what breaks down this blood-brain barrier? Gareth's lab and others have really studied the role of astrocytes in modulating the blood-brain barrier, the tight junction bonds between capillary cells. And so in two recent papers, one in 2012 and one in 2015, these two studies characterized diffusible signals that astrocytes produce to open up the blood-brain barrier. So what we do know is that when there's inflammation, microglia start to secrete a cytokine called interleukin-1, and interleukin-1 tells astrocytes to themselves secrete soluble signals. Two of these signals, one's called vascular endothelial growth factor A, and one is thymidine phosphorylase, which then leads to the synthesis of a soluble factor D-deoxyribose. So D-deoxyribose and vascular endothelial growth factor A diffuse from the astrocytes, and they bind to the endothelial cells of the capillary wall and tell the capillary wall to downregulate tight junctions. So the capillary opens up, and that allows for this inflammation to occur. Interestingly, also in conditions of non-inflammation, just at normal developmental static conditions, astrocytes are secreting another signal called sonic hedgehog. And sonic hedgehog tells the capillary cells to maintain their tight junctions. So there's this yin-yang push-pull where astrocytes, it can be thought of as a master cell that are telling the capillary wall to stay shut, and under conditions of inflammation, it then tells the capillary wall to open up. And so we understand that astrocytes are a really important regulator of the capillary wall, of the traditional blood-brain barrier. But this led to a further question: are these reactive astrocytes, are they good or bad? And are they protective or pathogenic because they're secreting all of this bad stuff that's opening up the capillary wall and allowing inflammation to happen? But in chronic conditions, they're also kind of maintaining this blood-brain barrier. So this is a picture from a review by Mike Safroniev at UCLA, and this also shows the neurovascular unit just in cross-section. So what you can appreciate here is, so you have the bloodstream, you can see the capillary cells, and those bow tie symbols are the tight junctions, which are the structural barrier. And then you have the astrocytic end feet processes, and then you have the brain parenchyma, which is the neurons and the astrocytes right there. So do we really need the reactive astrocytes? They seem like a bad player. So what was really unexpected and surprising was a series of studies that showed that the reactive astrocytes are actually protective. So in two separate studies, they were able to induce astrocyte cell death under conditions of inflammation. So the astrocytes were there present, and then when there was inflammation, they had two different methods of inducing astrocyte death. And when you eliminate the reactive astrocytes, you actually get worse inflammatory lesions. So there's something protective about the astrocytes themselves. Even though they're sending out these horrible chemical signals, there's something protective about them. And what we hypothesized was that maybe they were playing a structural role, that structurally they're acting as a second barrier around the classical blood-brain barrier. And so here's a figure of what happens when you knock out these astrocytes. And essentially, you don't have that second layer there around the capillary wall. And so inflammatory cells can seep into the brain and the spinal cord and attack the parenchyma. So here's where we get to the two barriers. So under healthy conditions, we know that the vascular endothelial wall is tight. It's expressing tight junctions, which are made up of a molecule called Cldn4 and Cldns and JAMs. And the astrocytes are kind of hanging out surrounding the capillary wall. When you get inflammation, what we hypothesized is that the astrocytes are sending signals to open up the blood-brain barrier, but maybe they're playing some structural role around the capillary wall. And since the paradigmatic molecular component that mediates structural barriers are tight junction molecules, we looked for tight junction molecules in reactive astrocytes. And so that's what we found. We looked both in vitro at human astrocytes that were exposed to the cytokine interleukin-1 beta, and we also looked into animal models of inflammation. And what we found, this is from the cultured cell data, is that under conditions of interleukin-1 beta, astrocytes express three tight junction molecules, Cldn4, Cldn1, and JAM-A. And these are particular members of the tight junction families, but they're unique to the astrocytes. So like I mentioned before, the capillary wall is expressing Cldn4, JAM-A, and occludin. So it's a different combination of tight junction molecules. And this combination is found in skin and bladder epithelium. So there's precedence for these three. This is a time course where we're showing that Cldn4 is induced by 24 hours, and Cldn1 as well. Other tight junction associated molecules stay unchanged, like zonooccludins 1 and zonooccludins 2. Occludin actually goes down with treatment of interleukin 1 beta. And this is an electron microscopy image where on the top panel we see an astrocyte cracked open. And under control conditions, astrocytes actually express gap junctions. So these are junctions that electrically couple the cells together. So astrocytes are communicating electrically with one another under control conditions. And then on the bottom, when you treat them with interleukin 1 beta, that inflammatory cytokine, they downregulate the gap junctions and they upregulate tight junctions. So they stop being electrically coupled and they actually seal up. So it's kind of a fascinating process if you imagine when there's an inflammatory insult, astrocytes, which are usually freely communicating with each other via these gap junctions, they seal off from one another, almost to protect the area that's being inflamed. And then this is another electron microscopy section where we stained for JAM-A and Cldn4, and you can see that it's present in the tight junction itself. So these are in vitro cultures of cells, of cultured human astrocytes. When you treat them with just a vehicle control, you see that they don't express Cldn1, Cldn4, and JAM-A. These are the tight junction molecules. So under control, regular conditions, they're not expressing tight junctions. The blue is the nucleus, and the green is GFAP. That's marking them as astrocytes. But when you treat them with interleukin 1 beta, you see an upregulation of these three tight junction proteins. Inflammation is inducing tight junction expression in astrocytes. So we wanted to test what the functional consequences of these tight junctions are. And the tool that we used was a conditional astrocytic knockout of two of these tight junction molecules. And we started off with Cldn4 because that's the most straightforward kind of structural tight junction protein. And the way we knocked it out was we had a Cre recombinase system where GFAP was driving an enzyme that would pull out the Cldn4 exclusively from astrocytes under inflammatory conditions. And what I'm showing, sorry, at the top, this is just under, these are regular animals being exposed to an inflammatory CNS model. And here, Cldn4 is being expressed here in the astrocytic end feet. And then we use these two knockout mouse models. Here in the bottom, this is labeled by MGFAP Cre Cldn4 floxed mouse and the MGFAP Cre JAM-A floxed mouse. And here we're showing that the Cldn4 conditional astrocyte knockout is not expressing Cldn4 under conditions of inflammation. So this was the tool we used to study how inflammatory lesions would look when you don't have this compensatory tight junction barrier forming. So the first model we used for inflammatory CNS lesion induction was a model where we inject an adenovirus expressing this inflammatory cytokine interleukin 1 beta. And we inject it into the cortex of control and astrocytic knockout mice, and then we wait 7 to 14 days, and we look at the lesions, and we compare the lesion size, the cell entry into the parenchyma, the soluble factor entry, the size of activated astrocytes, and we can characterize how these lesions look. So this is a model of inflammatory cortical lesions of the brain, and it primarily involves gray matter damage. So in our Cldn4 knockouts, what we found was that there was increased T cell infiltration in lesions. So what you're seeing here is the injection site of the adenovirus expressing IL-1, and you get more extravasation of CD4 positive T cells. You also get more staining of CD11b, which is a marker for macrophages, which are antigen-presenting cells and also damaging inflammatory cells. Here you see there's increased extravasation of IgG, which is immunoglobulin. Basically, all levels of antibody are escaping into the lesion. And we also looked at fibrinogen, which is a soluble protein in the bloodstream, which has recently been shown to act as a macrophage activator, and fibrinogen is also increased in lesions. We stained for NEUN, which is a marker for neurons, and this gives us a sense of the size of the lesion and cell death in lesions. And in Cldn4 knockouts, as you can see, the lesions are more exuberant and larger. As a control, we looked at Cldn4, which is the tight junction molecule that's expressed in the classical blood-brain barrier, and it was not different between our controls and our Cldn4 knockouts, which was what we would have expected. So the classical blood-brain barrier is unchanged in our Cldn4 knockouts. The second model of inflammatory CNS disease we used was a paradigm that's well established in the MS research world. It's called experimental autoimmune encephalomyelitis. And essentially, it's a way to induce a relapsing autoimmune inflammatory process like MS in live mice. And so it involves injecting mice with a protein moiety and then also an immune adjuvant, and that leads to an autoimmune attack in the spinal cord. And we score mice over the course of 21 days in terms of their motor function. And so here at the bottom, you can see the scoring system. At a score of around 2 to 3, the mouse is paraplegic. Around scores of 4 and 5, they're tetraplegic or quadriplegic, and they're sacrificed after scoring 4 for more than 2 days. So in our Cldn4 knockout mice, we found that the animals had more severe courses of EAE, and they had a higher mortality. And this is reflective of the fact that we think that the lesions are larger, they're more exuberant, there's less protection from this compensatory tight junction barrier that the astrocytes would have been producing. So here are the values for peak score and mortality in these animals. Work in progress is we're actually looking at the sections of the EAE lesions, and again looking at T cell infiltration into the spinal cord lesions. And here we see that again in the EAE, like the cortical injections, there's more T cell infiltration. There's also more fibrinogen leakage from the bloodstream into the spinal cord. So what's happening exactly with Cldn4 and JAM-A and all these tight junction molecules? In other tissues like gut, skin, we know that tight junctions are anchored to a cytoskeletal complex, and this can vary and lend specificity of function in different tissues. So it's known that claudins have a very characteristic complex formation that's different from the junctional adhesion molecules. So this is just illustrated here, where you see claudins are anchored to a molecule inside the cell cytoskeleton called Z01, and then it's all the way linked to the actin cytoskeleton. So it's really providing a strong structural foundation linking this membrane bond to the actual cytoskeleton of the cell. And JAM-A is known to have different binding partners. So what we did was we took cultured astrocytes and we pulled down Cldn4 and JAM-A, and then we did Western blots for various different adaptor proteins and looked at the patterns of the proteins that were bound to Cldn4 and bound to JAM-A. And this is a busy slide, but the basic point here is that Cldn4 pulled down with a unique combination of proteins that was different from the combination that we pulled down with JAM-A. And this is precedented from what we find in other tissues. Next, we wanted to see a little bit more in detail in vitro what happens when you knock out Cldn4 or JAM-A to the interactions of astrocytes with lymphocytes. So we developed this culture model system where we co-culture activated astrocytes with activated T cells. And we looked at the behaviors, the interactions between the astrocytes and the T cells under control conditions and when we knock out these 3 tight junction molecules. And the basic point I want you just to notice from this slide is that in the left column under the control conditions, astrocytes, when they're activated, they form these long processes that sort of link up together. And you can see they're kind of looped right here. And the lymphocytes are kind of corralled into clusters. And whether that's because they're binding to the astrocytes themselves or that they're just physically limited from diffusing because they're in this space is something we're trying to figure out. So when you knock out Cldn1, Cldn4, and JAM-A, the astrocytes are no longer binding to one another with those tight junction bonds, and the lymphocytes are more dispersed. And so we quantified that, and essentially when you knock out any of these 3 tight junction molecules, you get more lymphocyte dispersion and less clustering. So the next step of this is to figure out what astrocytes and lymphocytes are doing together in culture and what happens when you knock out these tight junction molecules. Are their gene expression patterns changing? Are they expressing different intracellular signaling pathways? Is this mediated through leukocyte adhesion molecule expression? So the next question we wanted to ask was, okay, so if in an inflammatory lesion, if the blood-brain barrier is opening up, but you get a compensatory close down of the astrocytes around the blood-brain barrier, how do the lymphocytes then get into the parenchyma to do their damage? Because we know typically they do get past the astrocytic barrier. And we hypothesized that lymphocytes, as they're known to do, express proteases and that they may be degrading the tight junction molecule and being able to exit into the brain and spinal cord parenchyma. And this is a figure of, this is simply an image from an EAE spinal cord showing lymphocytes that are really limited to the blood vessel perivascular lumen, and then they're actually breaking open into the parenchymal space. So what we did was we, in our co-culture system, we harvested the astrocytes under control conditions when they're treated with interleukin-1 beta, when they're treated with interleukin-1 beta and co-cultured with T cells, and then we looked for Cldn4 and its degradation product. And what we found was when astrocytes are co-cultured with T lymphocytes, over time, the Cldn4 begins to get degraded. And so we think this is the mechanism by which lymphocytes ultimately get through the second astrocytic barrier and attack the CNS parenchyma. And our next experiment is to see if we can block this degradation with protease inhibitors. So just to sum up, what we found here is that astrocytes, when activated by inflammatory stimuli like interleukin-1 beta, form a protective tight junction barrier. And so this is where A Tale of Two Barriers comes in. I don't know if you guys remember Tale of Two Cities from high school. I actually read it recently in residency while I was traveling to Elmhurst. But the basic story involves an upstanding French aristocratic citizen who does everything right, and he's married to a wonderful wife and is a responsible citizen. And so I would say he's kind of like the blood-brain barrier. He's protecting the brain from any kind of badness. He's doing his job. There's a second character who looks exactly like him. He's almost a physical double, who is a dissolute English drunken lawyer who doesn't do anything right, and he's in love with the good man's wife. But he really doesn't help society at all, and he's kind of like the astrocytes here. That's open. He's just hanging out. At the end of the novel, the good French nobleman gets imprisoned by the French Revolution, and he's going to the guillotine, and this is horrible. His family is distraught, and at the last minute, the dissolute English double decides to switch places with him and sacrifice himself so that the nobleman can get away. And so I think this is what happens under conditions of stress and inflammation. The good guy, the blood-brain barrier, has weakened and opened up, but the astrocytic barrier steps up to the plate and seals up and does its part to protect the brain. And you can see here the lymphocytes and immunoglobulins and soluble factors are escaping, trying to get into the CNS parenchyma, but they're held back just a little bit. So this is not the full story. Our next steps are to look at the role of JAM-A, junctional adhesion molecule A. This is a second tight junction molecule, which is more complex and really exciting. It's different from Cldn4. What it's been shown in previous work is that JAM-A acts both as a tight junction structural molecule and as a leukocyte adhesion molecule. So in other tissues, even in brain vascular endothelium, JAM-A can start in the tight junction, then it can exit the tight junction and act to tag leukocytes and bring them into the CNS. And so we're wondering, is it playing a similar role here with the astrocytes? And so JAM-A may have both a protective and a pathogenic function if it's bringing in leukocytes. And we have the astrocytic JAM-A knockout mice to kind of explore this question. More broadly speaking, studying the blood-brain barrier and the astrocytic barrier points us to some very compelling therapeutic targets, not just for MS, but for other conditions where the blood-brain barrier is compromised. These include ischemic stroke, neuro-oncologic conditions, cognitive decline and aging. They all implicate breakdown of these barriers. So just speaking about MS, we have a lot of therapies that target T-cell trafficking and actually modulating T-cell entry into the CNS, but methods are really still in their infancy to reinforce the traditional blood-brain barrier at the capillary wall. There are really no tested therapies that are modulating the astrocytic barrier. There are some treatments that are looking at actually modulating astrocyte signaling to the blood-brain barrier. Gareth was involved in a phase 1 trial of bevacizumab, which is a VEGF-A blocker, which blocks one of those signals that opens up the blood-brain barrier. And then people are working on ways to promote myelin repair and prevent neurodegeneration. So in conclusion, just to sum up, what we found is that these supporting cells, astrocytes, form tight junctions in response to inflammation, and this is effectively a second barrier that's distal to the traditional blood-brain barrier of the blood vessel wall. And Cldn4 is one of the components of those tight junctions, and it's key to protecting against leukocyte and soluble factor entry. And when you don't have Cldn4, you have large inflammatory lesions and severe lesions that correlate to clinical disability. And the significance of understanding these two barriers is that it gives us more checkpoints in terms of controlling CNS entry of inflammatory factors. So there are essentially two barriers as opposed to one. This, as I discussed, is relevant to a wide range of CNS conditions, and it's also relevant for getting drugs actually into the brain that we want to enter the brain. So the future directions of this work are what the downstream intracellular pathways of these various tight junction molecules are. I mentioned the really intriguing possibilities for JAM-A in astrocytic function. We're also initiating a collaboration with Dratan Agaliu at Columbia, looking at 2-photon imaging of leukocyte behaviors in the spinal cord of these knockout mice to see how leukocytes behave and traffic into the parenchyma, whether it's different when you don't have these tight junction molecules at the astrocytic barrier. And then lastly, we discussed how these mechanisms can be applied towards therapy. So I want to acknowledge Candice Shapuli. She's really been my partner on this project and has taught me a lot, and it's been great working with her. Anthony Throttle is my trusty technician, and he's his salary's been provided for by the Leon Levy Foundation. And I thank Gareth for being an incredible mentor, and the Leon Levy Foundation for supporting this work. Thanks so much.
Hi, Sam. Very nice
Hi.
talk. You alluded to, on your fourth bullet, in significance in terms of drug delivery. So there are times we would want to breach the blood-brain barrier, not only for drugs, but potentially to enhance immune surveillance. Have you given any thoughts as to triggering, turning it on, turning it off?
Yeah, some of those molecules that were studied in earlier studies, including D-deoxyribose, that's a soluble factor that gets into the bloodstream through the GI tract. And this paper in Brain from last year, we looked at that as a factor for opening up the blood-brain barrier. When you block it, it prevents lesion severity. So I think that's a really tantalizing candidate in terms of something that you could, as an adjunctive, that could possibly help open up the blood-brain barrier for drug delivery. So there are lots of examples of that, and it's like the yin-yang of infection and autoimmune disease. There's also the yin-yang of opening up the blood-brain barrier and closing it down. And so I think these mechanisms are relevant to opening it up if we want to get things through the barrier as well.
Next question.
Here. Thank you, Sam. Very interesting work. I wanted to ask you, since you showed that these tie junctions, these astrocytic tie junctions, are increased in inflammation, what do you think, what would you expect to happen in a disease such as Alzheimer's, where there is also a toxic factor, so the astrocytes have to deal with amyloid and probably caspase activation, death receptor activation, and a lot of other neurotoxic factors going on?
Yeah, I think it's a totally fascinating area. Astrocytes do a lot of things. Just in the ways they modulate the blood-brain barrier, they can do that bidirectionally. And so on a chronic time frame, there are a lot of different pathways implicated, but it is of relevance. Even in MS, we know that there's chronic activation of there's reactive astrogliosis in old lesions of progressive disease. And so understanding what pathways are active in astrocytes at different stages in the acute inflammatory setting, but also in the chronic inflammatory setting in contexts like dementia are important. And, I think nothing is all one or the other. I think they're both pathogenic and protective pathways that these astrocytes are involved in.
Transcranial electrical stimulation has been claimed to be useful for a variety of different diseases of the brain. Mm-hmm. Nobody has a clue how it works. Recently, Hajime Hirase's group have looked at how DC stimulation transcranially affects astrocytes, and he has seen extraordinarily big calcium waves traveling in the cortex. So one question is, can you speculate how astrocytes can feel electrical fields? The second one is whether this kind of a field stimulation outside the brain can be used for autoimmune diseases or opening the blood-brain barrier or potentially for other ailments.
Yeah, I don't know enough electrophysiologically how transcranial magnetic stimulation modulates ion channels and conduction, but you can imagine if they're communicating by gap junctions that there may be a mechanism for them to be perpetuating an electrical signal. But I think I would just be speculating on how it might affect an autoimmune process. I don't know enough about it. Sorry.
Over here.
Hi. I'm wondering about our English astrocyte friends and why they're so dissolute most of the time,
Yeah.
and if there's a role for that in terms of regulating the tension between astrocytes in a positive way, besides just being permissive for destructive and invasive immune processes. And one thought that had crossed my mind, but please speak more broadly, is in terms of vasoconstricting and dilatory processes in terms of modulating cerebral blood flow. And if the astrocytes are able to actually flexibly and adaptively compensate the tension between them so as to facilitate vasodilatory processes.
So I know there's a vast literature on astrocytes modulating cerebral regulation. Again, that how to modulate those processes and how that would be applied to an inflammatory process. I'd have to look more into, but definitely I think it is, like I mentioned earlier, astrocytes throughout development and at baseline are secreting signals like sonic hedgehog that are maintaining the blood-brain barrier. So they're also performing positive maintenance and vascular homeostatic functions. So they're not lazy, they're not dissolute.
Yeah, I was just going to ask you about sonic hedgehog. It doesn't interfere with any of the kind of things you have looked at?
I'm sorry, in what way? Sonic hedgehog, in what
Yeah,
way?
yeah.
Oh, whether if you
You mentioned that
modulate
it's a protected device, but
yeah.
but you never mentioned it again. So I was wondering.
Yeah, so that was the work from Alexandra Pratt's lab, the sonic hedgehog work. But I think it is an experiment begging to be done to see if you can add sonic hedgehog pathway modulation with some of these other pathways like VEGF-A and DDR to see if you get even more, if you can enhance the protective effects, because you never fully protect against inflammatory lesions when you just block or treat one pathway. But yeah, I think doing a combinatorial effect would be interesting to do.
So one last one, so that I protect our break time for, yeah.
I was just wondering what you thought of in paraneoplastic syndromes that might be happening, like NMDA receptor encephalitis, how antibodies might be crossing the blood-brain barrier and what may be making it permeable in those conditions?
Yeah, so I can speak a little bit to that. So like in conditions like neuromyelitis optica, the antigen is actually on the astrocytes themselves. And so so the actual pathway by which the blood-brain barrier opens up and then these pathogenic antibodies get past that and then past the perivascular space and past the astrocytic barrier is part and parcel of what we're looking into, how the astrocytic barrier opens up. But you can imagine if you're attacking the astrocytic end feet themselves, you're damaging that structural layer, and so they could get into the parenchyma. NMDA receptor is, the target is on the neurons, so how it actually gets past, it's still, I think, a part of examining what the pathogenesis of the barrier opening is. There may be a role for resident B cells secreting their antibodies within the CNS. So a number of possibilities.
Thank you very much.
Yeah.